note from the organizers there. I guess I can go ahead and get started. So thanks everyone for joining. My name is Cory Suski. I'm here at the University of Illinois in Champaign, Illinois, and I'm going to share some of the work that my group has been doing, developing new barriers to stop the movement of invasive Asian carp. So I want to say thank you to everyone for joining, and thank you to the organizers for inviting me to present. And yeah, I'm really looking forward to sharing some of the work we've been doing. We've been doing this for a few years now, and so we have a bunch of different studies that we've kind of gone through and kind of put together. So I'm not going to have time to go through everything today and do everything in a lot of detail, but hopefully gives you an idea of some of the work that we have been doing. And I have a bunch of the papers and a lot of the resources available on my website if you're interested to go grab those and look at those in a little bit more detail. So the story today is about Asian carp, and that's a aquatic invasive species. So sort of the umbrella where everything kind of sits is relates to this topic of aquatic invasive species. And they're very common around the world. There's a number of different examples of invasive species that have been spread around the planet. That's pictures at the top there of some zebra mussels. They spread, they proliferate once they get to a new area. A lot of this is fueled by global trade and commerce, so things like shipping vessels like that I've got on the slide there. That's what is having helping animals move around the planet. And the reality is, when you look at invasive species, especially aquatic invasive species, is once the organisms show up in a new area and start to spread and produce new babies and those kinds of things, it's very difficult to get rid of them. So once something shows up and becomes established, it's basically there forever. Removing them is very expensive, very difficult. And so the key when it comes to aquatic invasive species is to prevent them from showing up in the first place. Once they're there, it's tough to get rid of them. So prevention is kind of the key for them to, in terms of control. And the Great Lakes region, I mean, is a particularly prime example of invasive species and some of their impacts. And this figure here is from a paper from a few years ago, but it shows the cumulative numbers of invasive species that have shown up in the Great Lakes going back to about 1840. And so you see the total just keeps going up and up and up over the past little while. So altogether, creeping up on 200 invasive species that have been in the Great Lakes over the past 100 and some years. That translates to approximately a new species showing up in the Great Lakes every 28 weeks. So they're kind of a hotbed for invasive species showing up, partly due to commerce and travel and trade and all those kinds of things. And so it's been a particularly problematic area. There's a number of reasons why invasive species are bad. A big one is cost and the financial aspect of having these invasive species showing up. In some of the papers that I have there, invasive species are believed to cost someplace between $1 and $5 billion in terms of trying to clean them up, get rid of them, control them, mitigate some of their negative impacts. We spend millions of dollars, for example, on control for lamprey. 88 species of introduced mollusks result in about $1.7 billion in costs. And so again, just to reiterate, to prevent the key is to prevent these species from showing up in the first place. Once they're there, we got to deal with them, tough to get rid of them. So prevention is kind of the key to keeping some of these costs down. And in addition to some of the financial impacts of having these animals show up, there's problems with biodiversity, and biodiversity loss. And the reality is that when you look at freshwater ecosystems, they arguably are the most threatened ecosystems in the world. That yellow figure there that I have on the bottom is loss of biodiversity. So this is kind of the numbers of organisms and sort of their genetic diversity and their functional diversity, and basically just like how species rich and functionally rich a particular ecosystem is. And you see that over time there in that yellow figure, there's been a decline of diversity across the planet, with the green areas being terrestrial areas, the light blue being marine areas, and the purple areas or darker blue being freshwater environments. So freshwater environments, we're losing biodiversity, and we're losing biodiversity to freshwater environments at arguably a faster pace than the freshwater or the terrestrial or the marine environment. And non-native species are part of the problems when it comes to this loss of biodiversity. And this is something we want to prevent and avoid having happen. And so invasive species kind of factor in with that. And that was sort of biodiversity broadly within freshwaters. But if we look at just at freshwater fishes and some of the problems of freshwater fish around the planet, we see that freshwater fishes are among the most threatened group of organisms on the planet, sort of after amphibians, or maybe in worse shape, but fish aren't too far behind them. There's a bunch of people that have looked at this in terms of like quantifying invasive species around the planet, and about almost 40% of described North American fishes are currently imperiled. And from 1989 to 2008, the number of imperiled fishes in North America actually increased by 92%. So the more and more fish are becoming threatened every year, and things are only getting worse for fish. And again, extinction rates of freshwater fishes are believed to exceed that of terrestrial fauna. So freshwater fish are broadly, biodiversity in freshwaters is in a precipitous condition, I guess. And freshwater fish in particular are feeling the impacts of a lot of different problems, in particular invasive species. So now we can focus a little bit more on carp and the story, the research that we've been doing when it comes to carp. And with a little bit more background, there's two main sort of sides to the story when we talk about carp and what's going on with carp specifically in the Mississippi Basin. And the two players, I guess, are the Asian carp themselves and then the Chicago area waterway. The Asian carp side of the story is relatively straightforward. In that Asian carp, there's two main carp, big-headed carp. There's the silver carp on the left and the big-headed carp on the right, and those are the two ones that people are the most concerned about at the moment. That may change in a few years as other species begin to proliferate or could proliferate. But these used to be their filter feeders, so they swim around basically with their mouths open, and the best way to kind of describe it is that these species of carp will eat when they breathe. So they bring water in and they filter water through their gills, and they eat the small particles that are in the water. And so they're really good. They were brought to North America intentionally to clean aquaculture ponds and to clean sort of like sewage treatment lagoons, because they're very, very good at swimming around and cleaning out the water because they eat when they breathe, which is sort of a weird concept. And it was sort of the middle part of the Mississippi. The story, the tale that's told is that they were in Arkansas. There was a flood. The Mississippi River and the Mississippi River flooded, and the animals escaped out of their aquaculture pond and got into the Mississippi River, and then they've spread ever since then throughout the rest of the Mississippi Basin. They're heading north and south up and down the Mississippi, and they're getting precariously close to going into the Great Lakes. They're not actually in the Great Lakes. They're not super close to going into the Great Lakes, but they're getting closer. The reason we don't like this species of fish, there's a number of reasons. They've obviously spread very rapidly. They're very, very abundant, fast growing. They can have up to 2 million eggs, and they spawn multiple times per year. And they compete with native species for food. So they are filter feeders. They eat phytoplankton and zooplankton out of the water column, and all baby fish, all native baby fish will also eat phytoplankton and zooplankton out of the water column. So these invasive carp are then competing with native species for food. And if you haven't done so already, I've got some pictures there, but go to YouTube and type in Asian carp, jumping carp, that search on those types of words, and you'll see videos that represent some of the pictures that I have on the screen there, where there'll be hundreds, if not thousands, of fish simultaneously in the air at once. So they've become, basically, in a lot of areas in the Illinois River, the Asian carp have become the most abundant fish in that part of the river. So they're negatively impacting the native communities. They can jump in people's boats and break their fishing rods and those kinds of things. So they're kind of a nuisance, and they're not something that we want to spread around. Then again, controlling them, preventing them from spread is the key. Because once they're here now, it's tough to get them to go away. The second part of the story then deals with what's called the Chicago Area Waterway, the cause, or the Chicago Sanitary and Ship Canal, the CSSC. So there's a bunch of different names for it, but it's basically an artificial channel now that goes through the city of Chicago. So a long time ago, hundreds of years ago, the Mississippi Basin and the Great Lakes Basin were separate. And so there's, as you see in the top figure there, there's a little yellow dotted line, and that's a little baby continental divide. So it's a little sort of it's a little sort of hill, I guess, that runs along that yellow line. And anything to the west of that yellow line is part of the Mississippi Basin, and that water flows to the Mississippi. And anything to the east or to the right then is part of the Great Lakes Basin, and that flows into the Great Lakes. But the point is that there's a little hill there. There's a continental divide, and the Mississippi Basin and the Great Lakes Basin were separate. Previously, again, hundreds of years ago, Chicago used Lake Michigan both to obtain its drinking water and to dispose of its sewage waste, which is never really a good idea. And so to get around this problem, what the city of Chicago decided to do is to build canals and reverse some of the flows of some of the rivers, and to use the rivers and these canals then to dispose of its treated sewage effluent. So there's the Chicago River, which is right downtown Chicago with Navy Pier and all these sort of like iconic landmarks that are down there. That water, the south branch there, I can kind of, south branch, again, it flowed into Lake Michigan. Canals were dug. You look at the bottom figure now, you see the existing canals, and a bunch of canals were dug that go across that continental divide, and the water now flows from Lake Michigan across that continental divide down the Des Plaines River into the Illinois River into the Mississippi and down to the Gulf of Mexico. So the point now is that 78 miles of canals were built through that continental divide. There's pumps installed on the shores of Lake Michigan. We've reversed the flow of the water, and humans now have dug this artificial connection that flows between, allows water to flow from Lake Michigan into the Mississippi Basin where carp currently are. So what we have right now is this artificial canal that's been dug. It receives stormwater overflows from 740 square miles of urban area. It's responsible for getting rid of this treated sewage effluent from the city of Chicago. So it's a really important waterway from that perspective. But in addition, there's a lot of transport of goods and services that move up and down the Chicago area waterway, things like sand and scrap metal and coal and these kinds of things. So it's an important shipping avenue. It gets turned green in St. Patrick's Day, as you see there, so it's economic, or there's a lot of recreation that happens there. But the point is, this is an artificial connection now between the Mississippi Basin and the Great Lakes Basin, where carp could theoretically swim to go from the Mississippi into the Great Lakes and then off onto the rest of eastern North America. And if you recall from where I started at the beginning of the talk, the key for invasive species is preventing them from moving. Should carp get into the Great Lakes, it's going to be very difficult, if not impossible, to get rid of them. There's a number of ways people have been working to prevent carp from moving from the Mississippi Basin to the Great Lakes, one being an electrical barrier. So there's literally an electric fence, so it's not a physical fence. It's a fence made of electricity that supposedly would prevent carp from swimming from the Mississippi into the Great Lakes. The carp aren't anywhere near that fence, or about 30, 40 miles downstream, but this fence is effective. So, I mean, fish don't like that electric fence, but it might not be perfect. It's subject to power outage and maintenance, and there's potential for barges that move up and down the river to shield some of the fish, to allow them to pass. And in general, electricity is not that effective for small fish. So if we have small, and by small I mean sort of fingerling-sized fish, they get that electricity doesn't really work well for them. So the fence kind of works, but it's not perfect. And so we're looking for other alternatives then that can prevent carp from moving and prevent them from spreading into a new environment, ideally into the Great Lakes. So to look at this in a little bit more detail, there's something that came along a few years ago called the Great Lakes and Mississippi River Interbasin Study, the GLIMRIS report. As you can go look that up on the web. It's 400 or 500 pages long, and it's really kind of dry. But what it did was the GLIMRIS report looked at what can we do, what are the options, didn't make any recommendations, it just kind of surveyed options and said, hey, what can we do potentially to stop invasive species from moving from the Mississippi into the Great Lakes and from the Great Lakes into the Mississippi Basin? What are some of the options that are there? And those options, there's a range of options that are in that report, everything from doing nothing to building some walls to there's a bunch of different things that are in there. And a lot of them involve sort of this extensive and expensive replumbing of Chicago to change how the city of Chicago deals with its stormwater and deals with its treated sewage effluent. And here's an example of one of the potential options. I can't remember which one. I think there's 9 options or something like that. And so this is one of the options where they're going to change, put some physical barriers on the shores of Lake Michigan there at a Navy pier, and they're going to add some different water sewage treatment plants and basically make it so that things can't swim back and forth between those basins. As you can see in the bottom right corner there, that's an $18 billion price tag. I can't remember how long it would take. It would take 10 years or something like that to build. So it's very expensive to replumb a city. To build some new water treatment plants. It's very expensive. It takes a long, long time. And so there's a lot of potential obstacles there that need to be overcome if we want to stop carp from moving through up and down that basin. So where I start to fit in now is my group has been doing some research to try and determine if there's something that we can add to the water that would prevent carp from moving between those basins, but that would still allow water to flow back and forth, that would still allow that treated sewage effluent to leave, that would allow the stormwater to leave the city of Chicago so the city of Chicago wouldn't flood, and also that would allow sort of the barge traffic to continue, that would allow some of the goods and services, allow people to continue using the barge, turn it green on St. Patty's Day and all that kind of stuff. And so we're looking at what we're calling it, what can we put in the water that would act as a non-physical barrier, ideally a chemical barrier. And if you look at barriers broadly, these types of barriers, there's 2 different types of barriers. There's what's called a pull barrier, or we're attracting something. We're trying to get fish to not go in a particular area by pulling them to a desired area. Or there's what's called push or distractors, where we're trying to keep fish out of an area by pushing them away. Okay, none of these are going to be 100% effective, none of the push or pull barriers, but basically, the question we're asking is there something we can add to the water that would act as a push or pull non-physical barrier that would provide some redundancy with that electric barrier, some safety, supplement that electric barrier, and ideally address some of the problems with the electric barrier, especially when it comes to small fish and potential problems with small fish passing through the electricity. And so my group's been working with using carbon dioxide gas, so literally carbon dioxide gas, the same sort of thing that would be in soda, and seeing if that can be an effective non-physical barrier. There's some advantages to CO2 and there's some disadvantages to CO2. One of the advantages is that it's a naturally forming compound, meaning when we breathe out, we produce CO2, fish produce CO2, bacteria produce CO2. But the other problem with it is that it's a weak acid. It forms carbonic acid when you put it in the water. If you put CO2 into the water to prevent carp from moving, you will inadvertently and unavoidably produce carbonic acid. But the reality is that fish have an ability to sense CO2, and that's common across a lot of taxa, and fish have CO2 sensors in their gills, and so it's something that they can actually sense in the water. So the rest of my talk then, with that sort of the background, the rest of what I want to talk about today is some of the research that we've done where we're trying to determine the efficacy of carbon dioxide gas to act as a non-physical deterrent to influence fish movement. If we put CO2 into the water, can we keep fish from swimming from the Mississippi into the Great Lakes, prevent them from spreading and prevent them from establishing in a new environment? So the talk's going to have basically 3 sections. I'm going to talk about, first of all, how fish respond to CO2. Then I'm going to talk about some gene behavior and some work we've done with small fish, and talk about some whole animal behavior. And talk about avoidance of CO2, just sort of showing that fish do actually avoid CO2, not to give away the punchline. And then talk about some of the future work and ongoing work that we've got underway in the lab now. So one of the first things we looked at was just how do fish respond to CO2? Do they not, what happens if we take an animal and put it into high carbon dioxide environments? So the goal of the study was to quantify the impacts of CO2 exposure on larvae and on juveniles. So for juveniles, again, we're trying to address what I talked about previously with the fact that that electric barrier might not be super effective against small fish. And so there's a picture of the juveniles we're using. So they're just a few inches long. They're 2 to 3 inches long. And then that's the juveniles. And then the larvae, I don't know what you can see on your screen, but the top picture is a picture of a cup that actually contains silver carp larvae. So those are 8-day-old larvae. They're about the size, a little bit bigger than your eyelash, not much bigger than your eyelash, but they're really, really small. So we wanted to see how high CO2 would impact these organisms. We exposed these organisms to high CO2, and we quantify stress in the animals. And to do that, we looked at their genes, and there's different genes that are indicative of stress in fish. And so we looked at the patterns that these stress genes were showing after we put the animals into high CO2. So first figure that I'll show looks something like this, where we had the vertical axis here, the y-axis talks about a gene something called heat shock proteins and heat shock protein 70. And heat shock protein 70 basically is produced by an organism when its proteins start to break down. So when heat shock proteins go up, it means something is going wrong with your proteins, and your proteins aren't really functioning as they should, and so it's the body trying to correct problems with proteins. So what we have here is the amount, the expression of genes that code for heat shock protein 70. This is an 8-day-old bighead carp larvae, so they're about the size of your eyelash. We have these animals in high CO2 for either 30 minutes or 60 minutes. And then the black bars are control, so that's no CO2. And then we had 70 milligrams per liter CO2, and then 120 milligrams per liter CO2. So sort of 0 CO2, medium CO2, and high CO2. Now what you see is that if you take 8-day-old bighead carp larvae and you put them into either medium or high CO2, even for just 30 minutes, you get this upregulation of their HSP70 gene. So having carp larvae in high CO2, even just for 30 minutes, something goes wrong with their proteins, and their proteins just don't start to function well, and they produce heat shock protein 70. After 60 minutes, again, you see, especially at that high CO2, you see about a 3-fold increase in heat shock protein 70. So the animals don't like being in high CO2. Their proteins start to do weird things, and they really become unhappy with that. This is now looking at the juveniles. So juvenile silver carp, we took their gill tissue, and this is a gene called CFOS. And when CFOS turns on, it turns on all kinds of other genes. It's just sort of indicative of all kinds of problems with fish, basically. And again, we had 30 minutes or 60-minute exposure, animals at 0 CO2, 70 or 120 milligrams per liter CO2, and the same sort of thing was happening there. We've had juvenile silver carp looking at their gills, put them in high CO2, and they start to have problems with their gills, and their gills just basically aren't functioning as they should from being in that high CO2. So this is, again, with these small fish that are potentially vulnerable or not susceptible to the electrical barrier. And so this is good. We've got genes turning on in baby fish, showing stress and disturbance, and that was kind of cool. But that's really not going to stop fish from moving, and it's not really indicative of a barrier or something that would stop a fish from swimming from the Mississippi Basin into the Great Lakes. So we started changed a little bit of our focus and started looking at behavior and the behavioral responses of the fish to high CO2. And for this, what we did is we took bigger silver carp, like you see in that picture there. We put them in just in individual containers like we've got in the picture there, held the fish at 3 different CO2 concentrations for 3 hours, and every 15 minutes or so, we went and looked at the fish over that 3-hour period and just quantified its behavior, like what's it doing when it sits in high CO2 for 3 hours. And so the data looks something like this. So here we've got the y-axis here is the proportion of fish showing irregular behaviors. And so this is things like, is the fish twitching? Is the fish trying to jump out of its tank? Is the fish sort of gasping at the surface, having some breathing problems? Is it doing weird ventilations with its gills? So these types of irregular behaviors, okay? And so, and then we have our control animals at no CO2. Again, fish at 30 milligrams per liter CO2 and 70 milligrams per liter CO2. The black bars are largemouth bass. The light blue bars are bluegill, which are native species that would live in the Chicago area waterway. The red bars are silver carp, and the blue bars are bighead carp. And what you see is that at control environment, control concentrations on the left, there's very few animals in our study that are showing those irregular behaviors. But once you take an animal up to 70 milligrams per liter, almost everyone in the treatment is showing some sort of irregular behavior. And so this behavioral response basically mirrors what we were seeing with the gene data. Fish just don't like being in high CO2. They start gasping, they start twitching. You put them in 70 milligrams per liter, they're just unhappy, they're cranky, they want to get out of there basically. Next thing we looked at was what's called, we looked at ventilation. So we did for all the species, but I'm just showing the silver carp data here. So this is ventilations. So fish, when they breathe, they move their opercula, they're like this, and so they kind of basically sort of flap their opercula. And so we just counted opercular beats in ventilations per minute. And again, we had animals, there's silver carp, we had animals, the dark blue bars are control animals with no CO2. The light blue bars are animals at 30 milligrams per liter CO2, and then the dark blue bars are animals at 70 milligrams per liter CO2. So we had no low, medium, and high CO2. And these are the animals that are sitting in those containers, and we're monitoring them every half hour for 3 hours. And we start by looking at just the dark blue bars there. What you see is the dark blue bars don't change across any of those different time points. And so the animals, putting silver carp in a bucket of water does not change their ventilation rate. They ventilate at the same rate, just being in a bucket of water. Take that bucket of, then look at the light blue bars, and you take that bucket of water up to 30 milligrams per liter CO2. Again, there's no change in ventilation rates there. That's not a problem for them, and their ventilation rates don't change. Take that animal up to 70 milligrams per liter CO2, and we see the red bars all disappear. And the reason that the red bars disappear is because all those animals, all those carp basically passed out. They lost equilibrium and they became unconscious because high levels of CO2 is a fish anesthetic. The fish just literally went to sleep. So we couldn't collect any data from them. The animals went to sleep, so we scooped them out of their bucket, put them in a different tank, and they kind of woke up and they were fine. But the point is, again, this is just sort of driving home this point that CO2, fish don't like being in high CO2. It's a problem, it's really uncomfortable, and it is an anesthetic. And if we force them to remain in high CO2 for long enough, they will pass out. They will lose equilibrium, and they will go to sleep. So it's a very powerful compound. It's something they don't like, and it's really something that's has a lot of potential to act as a barrier. So to summarize then some of the behavioral data, Asian carp respond negatively to CO2. We can take those larvae and those fingerlings, so those 2 to 4 inch fish or those 8-day-old larvae, and put them in high CO2, and that activates their stress response, and proteins start to break down and proteins start to denature and those kinds of things, and they really don't. We see a physiological stress response. But extended exposures to CO2 will also result in irregular behaviors and ultimately equilibrium loss. It's a really powerful compound. Fish don't like being in high CO2. It's an anesthetic, and it's something that really has a strong impact on them and has potential to work as that non-physical barrier. So this is great. We can turn on stress genes, and we can maybe make the fish uncomfortable. We can even make them pass out. But it really still doesn't really get us that much closer to putting together a convincing argument to use CO2 as a barrier to stop movement. So now we changed it a little bit again, and we started to look at avoidance behaviors. So we're still looking at behavior, but we're looking at avoidance. And to quantify avoidance, what we use is this thing called a shuttle box. And so the picture, the bottom picture there shows the shuttle box in the lab, and the top is kind of a top-down view. So what this is, it's a computer-controlled choice arena. So we have two different chambers, and you can put a fish in there, and you can change the parameters of the water independently on either side of that arena. The fish can swim back and forth between the arenas, but there's very little water mixing that goes on between them. So you can see in that top figure, we get the pink side and the yellow side, and the two aren't really mixing. The pink water stays on the pink side, and the yellow water stays on the yellow side. So we can change those parameters, and the fish can choose where it wants to go, and the fish can let us know when it wants to get out of there. So what we did for this study was we put fish fill the shuttle box with water, put fish into one side, let it kind of become happy in there. And whatever side the fish was on, we just slowly start adding carbon dioxide. We let the carbon dioxide get higher and higher and higher and higher. And eventually the fish doesn't like it, and it swims to the other side. We can then take the carbon dioxide away from the first side and start adding carbon dioxide for the second side and chase the fish out of there and chase it back to the first side. So we can keep basically ping-ponging the fish back and forth from side to side. And as we do that, we can sort of write down when the fish leaves. And if you do that with lots of fish or with lots of different species, lots of individuals, you can get an idea of what would be avoidance thresholds where the animals want to leave CO2 and go to water. That would be more favorable. And so what these data here now show are adult fish in the lab, in the shuttle box. We got largemouth bass, bluegill, and silver carp, and we see the CO2 concentration that made them leave and swim to the other side of the tank. And what you see basically is that if you get, once CO2 gets up to around 100, 120 milligrams per liter of concentration, all the animals leave. There's some variation around that, and that's fine, but there's no difference across the species, meaning that a largemouth bass responded at the same level to bluegill that responded at the same level as silver carp. So all animals, fish don't, this just reinforces, fish don't like CO2, and at about 100, 120 milligrams per liter, adult fish will decide to swim away. We repeated this with the juvenile fish. These are the 2 to 4 inch individuals. Again, now with bluegill, largemouth bass, silver carp, and bighead carp. Same data, meaning fish swim away, but the concentrations were a little bit different. They need a little bit higher here, so maybe 150 to 200 milligrams per liter, but still. If you can get water up to around 150 to 200 milligrams per liter, fish will choose voluntarily to swim away from that area, and they won't because they don't like being there. Hopefully having CO2 function as a non-physical barrier to keep fish from an area that we don't want them to be in. Which is great. We can put fish in a tank in the lab and we can make them swim away, which is good. But again, the point of the research is to keep animals out of the Great Lakes through the Chicago area waterway. So the stuff we're doing in the lab in a tank is cool, but we need to get big. We need to get serious, and we need to take it up to a bigger scale. So that's what I'm going to talk about now is CO2 in a pond. Okay, so quantify the effectiveness of a zone of elevated CO2 to influence the movement of free-swimming Asian carp. We're going to go up to a pond. Now we're thinking rather than thinking about lab studies or lab simulations. Now we're thinking, well, let's maybe try and get this soon into the real world. Maybe get it into a river or into something kind of big. So what we did now is we got a pond. Some friends of mine that work in Wisconsin had a pond. It was about a half an acre pond, about 5.6 million liters. So I can't remember, 6 million gallons. So it's a big pond. So it's, we're looking at, a good sized thing here, coming up to the size of a football field, so to speak. And we had the CO2 infusion system where we had these fancy CO2 injectors basically, but we put them down at one end of the pond. And what we did was we monitored pH in the pond, because pH is like acidity, because remember when you put CO2 into water, it forms carbonic acid. So we used CO2 or pH as a proxy for the CO2. And we had telemetry, so we could look at fish positions as they swam around the pond. And so we tagged 20 tagged fish, put them in the pond, Gave them 22 hours to acclimate to the pond, to kind of get used to it, to find their way around. And then after 22 hours, we started adding carbon dioxide, but only to one end of the pond, not to the whole pond, just to one end, and left them for about 24 hours, drained the pond, filled it back up again, and repeated the whole thing. So we did the whole thing twice, looking at fish positions. Here's some pictures of what that looked like. So here's the pond. Top left one is the pond when it's drained. And then those white sort of sticks there is the CO2 infusion. So it's basically a bunch of just basically plastic pipes that carry water. We had a pump that would pull water out of the pond. The bottom left picture there is the CO2 infusion thing. And so that's where we'd add CO2 to the water. And then so the water gets pulled from the pond, goes through these pipes, gets treated with CO2, and goes back down and goes into the pond. And you can see the bottom right picture there is water being pumped into the pond after it's been treated with CO2. And then the top right picture is the plastic pipes. That's the CO2 infusion system just in one end of the pond. Here it is again, looking down the long axis. The pond's a big rectangle, so I was looking down the long axis of the pond. We're kind of filling it up. And the bottom picture is after the pond's been filled. So that's what it looks like. So we're looking at, again, it's about 6 million gallons of water, half an acre. It's a good size, real size pond. And so the data looked like this, and the best way to represent these data then I think is what's called a heat map here. And so the different colors represent the numbers of fish. So this rectangle, the rectangle represents the footprint of the pond. So that's the whole pond in rectangle. And then those black dots represent the CO2 infusion pipes that were down at one end, okay? So yeah, so that's looking from the top down at the pond, and then you see the area where we would be adding CO2 and what proportion of the pond that would be. And then the colors, the fish have all been moving around, and we took a 1 hour slice of time and we plotted out where we saw fish. And the fish locations are represented by the color. So the purple means high fish density, so there's lots and lots of fish there. And then we transition to the blue and the red and the green and then the red, and that means low fish density. So what you see is that this is the figure that I have up there shows the fish positions 11 hours before we added the CO2. So sort of during a control period, that's where what you see is the fish really like to be down at that. And they like swimming around all the sticks where the CO2 is. They like swimming around that area, and then they kind of like being up at the top end of the pond too. That's sort of the heat map would show that's where they like to be from the telemetry. And then again, we let them acclimate in there, and then we turn on the CO2. And so what happens then is we turn on the CO2, and we let that run for about 22 hours. And what you see is that 11 hours after we turn on the CO2, that's the distribution of the fish within that same pond. So it's a 1 hour slice of time. We basically plot all the fish positions for that 1 hour. And now what we see, we've kicked the fish out of the area where the CO2 infusion is. We made them all swim in the middle of the pond. So we've taken those animals where they're the preferred habitat in the control environment, and we've moved them and we've forced them to go now to the middle of the pond and not go to sort of their preferred environment. Then the last thing then is 20 hours after the CO2, the pond has now become a big soup of CO2, and the fish are all kind of swimming around. But the key figure there is the middle one, where we say fish like to be at the bottom end of the pond in the CO2 infusion, but we can kick them out of that area by turning on high CO2. So to summarize this part of the talk then, what I'm hoping to convince you with the avoidance data then is now CO2 really has potential to act as that non-physical push type barrier. Hopefully with CO2, we can keep fish out of an area. We can keep them out of their preferred habitat. The data that we've shown now with the avoidance is that free swimming fish will avoid areas of high CO2 once that target threshold is achieved. Once you get up to 100, 120 milligrams per liter, that's when we can keep fish out of an area. We've replicated that now in the lab, in that shuttle box, and in the pond. So hopefully those data look pretty good to you. It does not appear to be species specific, so largemouth bass, silver carp, bighead carp, bluegill, they all seem to respond the same way at about that same threshold. And this also does not appear to be size specific, meaning larval fish, juvenile fish, adult fish, they all seem to respond at about the same size. So I think it's just, again, part of this thing that I've been saying throughout the talk. Fish just don't like being in high CO2. High CO2 is just not comfortable for fish. Their proteins break down, stress axis turns on, and they just let me get out of here. This is terrible. So while it's really, really it has a lot of potential. I mean, I don't want to convince you or suggest that this is the answer to everything here. There's a lot of problems with CO2 and some of the stuff I've been showing, and that's sort of where we're working now and some of the future and ongoing work. CO2 is not a silver bullet that's going to just solve the carp problem overnight, and there's a lot of unanswered questions. For example, there's something called acclimation. So can fish just get used to high CO2? Like, it's fine to just take a fish, put it in high CO2 and say, well, I don't like this, but maybe they can get used to it. And so we put a CO2 barrier in place, maybe the fish will get used to it and then no longer becomes effective. There's the issue of environmental pollution. I'm sure people have all heard of this thing called climate change, and it's driven by carbon dioxide. And so do we really want to be putting lots and lots of carbon dioxide into the water, right? We know this is going to reduce pH in the waterway. We've shown that. What about impact to non-target organisms like native fish or mussels? There's a lot of organisms that way that could be impacted. And then how do you go about doing this? It's fine for me to say, oh yeah, just put CO2 in the water, but what's it going to cost? How do you do this? How do you pull this off? So that's sort of where I want to focus for the next part of my talk then, is talk about some of these types of issues and what we've been doing to try and address some of these questions to hopefully move this aspect of the research forward. The first thing I can talk about is acclimation. So again, do fish just get used to a high CO2 environment? Because if we put CO2 in an area and then the fish just get used to it, well, maybe it's not going to be effective anymore, and there's no point. So to test this, what we did is we had largemouth bass as our surrogate species because we had a problem with getting bighead and silver carp. Brought them into the lab, marked them all individually, so we know each individual. So basically, kind of gave them all names, and we got to know them all as individuals. Put them in, and we put them in the shuttle box, and we tested them all to quantify their avoidance threshold of each individual. Okay, so we know each individual fish's avoidance threshold. We then took them and put them in a tank like you see there on the top. We crank up the CO2 in that tank to really high, almost to the point where the fish are passing out, but not quite. Just back it off a little bit, and left them in that super high CO2 environment for 8 weeks. So we really give them a strong dose of CO2, and then we repeated our shuttle box testing to see what would change. The y-axis here, similar to what you've seen before, it's the concentration of CO2 that caused fish to swim away. The left bar is the concentration where animals would leave the first time we tested them, and then the right bar is where they would leave after 2 months of being at high CO2, and there's no difference across those bars. So basically, the line in the middle, there's about the average, the median in the middle. So it's about 180, 200 milligrams per liter. So there's no difference. So animals, there's no acclimation. So having fish sit at high CO2 even for 2 straight months with no escape did not change their avoidance threshold. So we don't see evidence of acclimation from high CO2. The sort of the pollution aspect of this, right? So when we add CO2 to water, we're going to drop the pH from the carbonic acid, and so we're going to have an acidification problem. The reality, though, is that carbonic acid is a weak acid, and it really doesn't get to. It's not like going to take the pH down to a pH of 1 or pH of 2. We usually, most of the time, at target concentrations, we see a pH of about 6.8. And a lot of environmental standards, including here in Illinois, as an example, we want to keep the pH. The regulations state that pH has to stay between 6.5 and 9, and that's sort of the comfy window that where things need to stay. And the carp barrier stays at 6.8. So really, there is a change in pH, but it's not a terribly large change in pH to the point where there's going to be a regulatory problem, at least in the water where we've been testing it so far. We did a study looking at how this is going to affect aquatic ecosystems. So there's the bottom left paper there is looking at biological consequences of weak acidification caused by elevated CO2 in freshwater ecosystems. So we reviewed, 150, 200 papers to see what's been going on. The paper just kind of came out, and, we're pretty comfortable with the impacts of high CO2 on individual species. Where we really don't know a lot is how high CO2 can affect ecosystems or the level of organization above an individual species. But in general, we don't see much of an impact or potential impact of pH change of this nature on individual species. That's usually within the pH tolerance ranges of individual species. That's why the regulations, for example, are to keep the pH between 6.5 and 9, because most animals are comfy in that area. And in terms of the climate change, global warming type aspect and CO2 production, all the work that we've been doing to date has used waste carbon dioxide that's been harvested from oil processing plants or soybean processing plants. So we're taking CO2 that would have been released into the atmosphere, that's being harvested, and it's being repurposed for these studies. And so we're not contributing excess CO2 to the environment. And so we're not contributing excess CO2 to the environment. Should the CO2 barrier be moved forward in the future, I mean, that's hopefully a source of CO2. We, that's our intent to kind of stay with that. But that's a little bit unknown at this point. But at least to this point, we're not like smelting anything and to generate more CO2 to run these studies. We're actually taking waste CO2 and repurposing it. I think the last thing I want to talk about in this section is impacts to non-target fishes. So it's fine, to have to do this work on the Asian carp, because that's our the invasive species we want to stop from spreading. But the reality is there's other species that are going to be in the water. They're going to be unavoidably exposed to a CO2 barrier should it be in place. We also know that there's a lot of work in the ocean environment on something called ocean acidification that's caused by elevated CO2 in the atmosphere that ultimately goes into the oceans. And the dynamics of that in freshwater are very different. But the point is that high CO2 in the atmosphere translates to high CO2 in the oceans. And there's been a lot of really cool research showing that that can have negative impacts on ocean fishes. They start to lose their ability to smell. The high CO2 in the ocean will cause those fish to lose their ability to avoid predators. They really become bold, and the baby fish will see a predator and they'll swim up to it, and so that can be really bad for getting eaten. And a lot of these mechanisms exist in fresh water. So we wanted to see if high CO2 could impact some of these behaviors in freshwater fishes, and see if there's some potential non-target impacts here that can spill over from a CO2 barrier to do bad things to some of the native fishes. And so what we had here, we use fathead minnows, which is a picture of a little baby fathead minnow there, because they have this thing called an alarm pheromone. And so they have a chemical in their skin, and when their skin gets broken, that chemical gets released into the water, and that then alerts their neighbors and their friends to say, hey, I just got kind of beat up and my skin has been broken and I've been attacked. Everybody get out of here, because it's basically a way it's an alarm pheromone, so alerts their kin that their skin's been broken. And so that's something that's been impaired in the marine environment when animals have been exposed to high CO2. So we wanted to see if that would happen with fathead minnows. So basically what we did here is we took fathead minnows, we held them at 3 different concentrations of CO2. We had ambient, or we had low CO2, or we had high CO2. So now we're measuring things in pressures where they're microatmospheres. So 600, 800, 7,000 microatmospheres. And again, when we're up at 7,000 microatmospheres, these animals are just about to pass out, and so it's really high CO2. We held these animals at this high CO2 for someplace between 4 and 11 days, and then we assessed their response to skin extracts from their conspecifics, and then we allowed them to recover for 11 days, and we repeated the testing. So we put everyone at high CO2, see how they respond to skin extracts, give them some time, put them back in low CO2 to let them recover, and then see again how they respond to skin extracts, and see if anything changes after 11 days of recovery. So to do this, we use this thing called a choice tank, and here's a sort of cartoon that shows what the choice tank looks like. And basically, it's a way to distribute water to fish to quantify their behavior. So we have 2 what's called header tanks that are at A and B there in the figure, and water from the header tanks just flows by gravity through a couple of pipes down through some flow straighteners, which are at section C, and then we get to section D where the fish is. So there's a picture of a fish at section D. So the water from tanks A and B then go to where the fish is at section D. And what we're able to do is put alarm pheromones into one of the 2 header tanks, either A or B, and then allow that and the water to go to the fish. So what we did, we put the fish in the choice tank here and give them about an hour to recover. Take a video of the fish before we give them skin extracts, which is how do fish behave in that tank. Switch the water flow and give them water that has the skin extracts in it, and then take another 10-minute video and video their behaviors. And one of the things we looked at were things like darts and freezes and how quickly they're swimming and how far they're moving. So basically, how do they respond once they've got this alarm pheromone from their conspecifics, and how does that change? So we get before skin extract and after skin extract, so the 2 different categories of data that we had here. So what we have here now is the data now on the y-axis here showing the number of darts, the numbers of the fish when they are darting around. So just kind of jumping around and say, oh, I don't like this and I'm really uncomfortable. So kind of do one of those. That's a dart. We're counting the number of times fish are darting. We had fish that were held at low ambient CO2, at low CO2 or high CO2. And then we had the before skin extracts are the green and the after skin extracts are the blue. And so we can look at the ambience. These are fish held at just regular water, right? And no carbon dioxide. And so what we see is that if we, before the skin extracts, the animals are hanging out, no one's really doing any darts. The darts are about 1, like they're pretty comfy. And then we give them the skin extracts, they start darting all over the place, and suddenly start darting 7 or 8 times during the trial. So that's what we'd expect to see, right? These are animals suddenly confronted with an alarm pheromone that is supposed to make them get upset and agitated, and that's what we see. So that's a good thing. For animals that have been held at low CO2, that response is still kind of there, but it's muted, right? So we've got the low, the before skin extracts, the animals are comfortable, and we give them the skin extract, they kind of respond. There's a bit of an increase in darts, but not really all that strong. And then animals that have been held at high CO2, we see that reversal. So before the skin extracts, those animals are jumping all over the place and kind of darting. Give them the skin extracts, and they kind of calm right down, and they don't really know what to do. And so the point is now high CO2 is altering the behavior of these fish, and they're no longer responding in a way that we would want them to after getting the skin extracts. But what happens then is these fish show recovery. So we take those same fish then and put them back in just an ambient CO2, leave them there for 11 days, and that behavior responds. So what we have here are the numbers of it's part of the data we've shown you before. So this is the numbers of darts that animals are showing after they've been exposed to high CO2, right? So after you've been exposed to high CO2 and acclimated to high CO2, we see high amounts of darts before skin extracts and low amounts of darts after. So we get this sort of odd behavior. Give those animals 10 or 11 days to recover in ambient CO2 and retest them, and their behaviors return. So now before skin extracts, they're really calm. After skin extracts, we have an increase in darts. We do see that this impaired behavior kind of goes away, and the animals start to behave a little bit more normally now after they've been given some time to recover. And the final sort of piece of this puzzle, things we're hopefully going to work on this summer, is just how to deploy this at a large scale. So again, we've been working in lab, or maybe this pond, this sort of half-acre pond, but the reality is if we want to stop carp from moving from the Mississippi Basin into the Great Lakes, we have to go deploy this into a river. So a big river is a very different song, so to speak, than it would be in a lab or in a nice tidy little pond in a research site. So we got to figure out how to do this. Cost, logistics. How do we do this? There's a bunch of different things we can do. You can have what we're calling like sort of a semi-permanent or a permanent deployment. We can put CO2 into a shipping lock, because locks are really nice choke points in the river. All the water kind of goes through a shipping lock, and all the fish can have to swim through the lock. And so if we can put CO2 into the lock, maybe we can keep fish from going into the lock and swimming through to the other side of the dam. That's something like semi-permanent. You might keep that in 4 or 5 months a year, all year. You don't really know what we can do. We got to look at the costs and all that kind of stuff. Or we can do what's called an intermittent or temporary deployment. We just put CO2 into one part of a river to keep fish from a spawning area, or keep fish from accessing food. Maybe we can just do that. Keep fish corralled into an area to facilitate harvest. So there's a bunch of different ways we can do that. And that's some of the things we're looking at in terms of this summer to just sort of work out some of the nuts and bolts and how to pull that off. The cost and logistics of deployment are still being developed, but that's going to depend on water flow, water chemistry, temperature, season. We know the amount of CO2 required will vary seasonally based on water temperature. So there's a bunch of other things we need to play with, but that's sort of what we're looking at this summer. So to start to kind of wrap things up here, hopefully I've convinced you that carbon dioxide gas has potential to act as a non-physical push-type barrier to help prevent the movement and establishment of bighead carp, to help keep them from moving from the Mississippi Basin into the Great Lakes, prevent their establishment, and hopefully keep the Great Lakes safe from their invasion. The data I've shown today hopefully convinced you that fish avoid zones of elevated carbon dioxide. Does not appear to be size specific, meaning juveniles and adults respond the same way. Does not appear to be species specific, so that's a good thing or a bad thing. So native fish respond the same way as invasive fish. We're looking at a target threshold of 50 to 70 milligrams per liter, about 60,000 microatmospheres in terms of pressure. And if we can get to that level, as far as we can tell, the animals decide to swim away on their own. Impacts to non-target organisms are possible, but appear to abate should the CO2 stress be removed. So we've seen that alarm pheromone, changing alarm pheromone behavior of our fathead minnows. But if animals swim away from CO2, that behavior is restored. And we have a lot of questions to work out in terms of logistics and cost and some of the impacts to the ecosystem level as opposed to an individual species level, and some of the deployment strategies. And that's hopefully what we're going to be working on this summer to try to move that forward to the next step. So in terms of wrapping things up, like I said, we've been doing this for a number of years now. It's a pretty big project. A lot of different collaborators from my group here at University of Illinois, USGS at the Upper Midwest Science Center, the Army Corps of Engineers, the Western Fisheries Center, and then the Illinois Department of Natural Resources, and Kevin Irons has been fabulous in terms of helping with things. So I'm happy to take questions. I see some questions popping up in the question box, so I can read that. So I can read the questions and I can hopefully try and answer them. So I don't know if everyone can see those or not. So would CO2 hurt other fish? Yeah. So from what we've seen, high CO2 is not species specific. So the invasive species and the native species would be responsive. A fish is a fish is a fish when it comes to responding to CO2. So potentially the CO2 barrier would have the same impacts on the native fishes as the non-native fishes. So they could, if we do put a CO2 barrier into a spot, that could potentially stop the movement of native species. So what that means then is we'd have to think carefully about, if a deployment does happen, where would it happen? What are the fish in that area? And what would happen if the fish wouldn't go through there? And maybe we need to think about that. Would this affect us as well? We don't see much of an impact on people. Again, it's not the CO2. There's more CO2 in your soda than there is in the in the water. To put that into perspective, so there's the change in pH is relatively small, and from the research we've done, we got to look at this a little bit more in terms of the CO2 getting into the air and impacting people nearby. It doesn't seem to be a really big problem. But we need to look at that a little bit more. If we left fish in the CO2 for a long time, they could die. They could. If fish are forced to remain in really high concentrations for a long time, yeah, they get really uncomfortable in that high CO2. Based on what we see, causing warming of water. We don't see any impact of CO2 on temperature of the water, but we do see, and the dynamics of CO2 in fresh water are very complicated. And atmospheric CO2 could play a role in CO2 in the fresh water in the water, and that probably is a factor, but it's one of many factors. That's a very complicated story. Someone say it sounds like a bad idea is initially introducing fish to the species. Yes, I mean there's a lot of reasons that why this could potentially cause problems. But I think it depends on your perspective on some of that too. If there's a lot of people that are in the Great Lakes right now that are saying, hey, we don't want these animals to show up in the Great Lakes. We need to stop them from moving. And so it's a really interesting dynamic when you bring in the sociology and the politics of it as well, in addition to the biology. And I think that's a really good point as well. So what group would pay for the implementation of the barrier? That's a really good question. I don't have to deal with that one. Yeah, I mean, because we don't really know. Because there's the installation and then there's the maintenance and the upkeep. And so one thing that you see with the electric barrier, there's maintenance that needs to happen. Things need to get cleaned, they need to be tidied, it needs to be repaired, and things break down. And so there's upkeep and maintenance. And yes, that's a really good question. And that's sort of something that is needs to be worked out. So will it contribute to global warming? Again, what we've done to date has been using waste carbon dioxide that gets harvested from soybean plants or oil and gas refineries. So to date, this technology has not produced any new carbon dioxide. It has been used carbon dioxide that would have been released anyhow, that would have been going into the environment, and it's been used just for this research and for these studies. I don't see any other questions, but I'm happy to take more questions. Hopefully I've answered all the questions. If there's something that I've missed, please remind me. Well, just curious where the pond was in Wisconsin. It was at the Upper Midwest Environmental Sciences Center in La Crosse. So it wasn't a public pond. It was a research site at the Upper Midwest Environmental Sciences Center. So technically it was a pond, but it wasn't like a public access pond. It was a research pond. All right. Well, I hopefully hearing no other questions. I hope people enjoyed the presentation. If you do have other questions, email me. Just search my name. Hopefully you can grab the slides and send me an email or whatever. Everything should be on my website, and there's a bunch of papers there too to go ahead and read those. And feel free to ask me questions. I'm happy to answer if I can. So thanks everyone for coming. Thanks also for the organizers for inviting me. I appreciate it. I like sharing the stuff and sharing the work that we've done. And so yeah, I appreciate the offer and the invitation. So thank you to everyone and have a great day. Thanks for attending the talk. Bye bye.